So thanks for the invitation to talk. I'm Thomas Graham, a postdoc in the Darzak lab at UC Berkeley. And today I'm going to tell you about a simple and inexpensive one-step RT-qPCR mastermix that our lab has developed for SARS-CoV. We experimented with a variety of homemade mixes over the last couple of months. And what worked best for us was just a simple combination of MMLV reverse transcriptase and Taq polymerase. It's really easy to purify these enzymes yourself, and a single liter of culture yields enough enzyme for about 40,000 to 50,000 10 microliter reactions. And we're happy to distribute these plasmids. If anyone wants them, you can email us at this email address. We tried a variety of different buffers and additives, and this is the one that worked best for us. So here's a standard curve with in vitro transcribed SARS-CoV-2 N-gene RNA using the N2 primer and probe set from the US CDC. We can reliably detect down to 50 to 100 RNA molecules per reaction, although we occasionally fail to see amplification at lower RNA concentrations. We were fortunate to obtain COVID-19 patient samples from a local healthcare provider. And while we were experimenting with homemade master mixes, we also experimented with simpler and cheaper RNA extraction protocols. And something that worked really well for us was just a simple isopropanol precipitation that involves adding linear acrylamide carrier, sodium acetate, and isopropanol directly to the swab sample. And this actually performed consistently better than a $6 per sample QIAGEN RNeasy kit. Here I'm showing you the CQ values for 12 different positive patient samples for 2 viral N-gene probes and a human RNase-P control probe. And the isopropanol precipitation gave consistently lower CQ values and better detection than the QIAGEN kit. In this experiment, we were using Thermo Fisher TaqPath master mix for RT-qPCR, but a couple of weeks later, after we had developed BEARMix, we tested the remainder of these samples using BEARMix. And although BEARMix failed to detect the 2 samples with the highest CQ values in the previous experiment, it did detect a majority of positive samples. And it's also worth noting that this experiment was done a couple of weeks after the previous experiment, and the samples were kept in storage. And so we're eager to repeat this experiment with fresh samples and test both of these master mixes side by side. One problem that we've observed with regular Taq polymerase is that it tends to form non-specific products like primer dimers after extended incubation at room temperature, and these can compete for the specific amplicon and lead to reduced amplification. So to address this problem, companies typically make hot start versions of Taq that only become active upon heating, and this usually involves including an anti-Taq monoclonal antibody, which is expensive. So we tried a less expensive approach, which involves reversibly inactivating Taq by formaldehyde cross-linking, and then incubation at 95 degrees for 10 minutes reverses the crosslinks and reactivates the enzyme within the reaction. And so we find that using this homemade hot start Taq makes it possible to keep reactions at room temperature for extended periods of time without compromising amplification. One limitation of this homemade hot start Taq, though, is that it gives less efficient amplification and higher CQ values, possibly due to incomplete reactivation of the enzyme. So this is something that we're still trying to optimize, and I'd welcome any suggestions you might have. So in summary, BEARMix can be easily assembled using homemade enzymes and is capable of detecting down to a few tens of RNAs per reaction. It's much less expensive than commercial master mixes, and we've shown that it can be used in combination with inexpensive isopropanol precipitation to detect viral RNA in actual patient samples. It's also possible to make your own hot start version of BEARMix, although the protocol may need some further optimization to improve amplification efficiency. So we'd like to thank all of these people who have contributed to this work. You can check out our protocol on GitLab, and please feel free to contact us if you have any questions or would like to send us, or would like us to send you our plasmids. And also, I'd just like to say that with everything that's happening in the news lately, all of the chaos and strife in the world, it's really encouraging for me and inspiring to see scientists around the world working together to address this common problem. So thanks to all of you for that.